So I'd like to thank everyone for coming today, and back in Delaware to be with you here. And I think my students are all working, so I don't think they're logging in, which is a good thing. But thanks to Mike Wilkins, who had done our last seminar, and a previous seminar by Brandon Briggs, we've received over 200 views on YouTube. And if you think about what the live audience was when things started, we had about 35 people per seminar. So we're hitting close to 300 people already, and I have to say we haven't really advertised. So this has been pretty exciting to think how broadly you can reach an audience with pretty easy effort and a freeware-based system. So today we'd like... Thank you, Kim. Today we'd like to welcome Deric Learman as a speaker and tell us about microbial redox reactions. And so Deric is coming to us from Central Michigan University, and he has a very, very studious office there with, actual whiteboard writing. I'm very impressed.
That's right.
And he had done his PhD in Virginia Tech and did his postdoc at Harvard and has headed to Central Michigan and is going to tell us about microbial redox reactions. And so, Deric, if you can say hi and share your screen.
Yep.
Yep. And just a reminder as we're sharing the screen that if you have questions, you can tweet them to the micro seminar tag. If you tweet them to Deric directly, he might not have enough bandwidth right now to be able to watch everything, so tweet them to micro seminar. We have not activated the Q&A option within Google Hangouts, and so the idea is if you're logged in, you can ask us directly, and if you're watching through YouTube, you can ask us through Twitter. So, Deric, if you want to take it away, you can share your screen, make it a full screen, and the rest of us will shut off our webcams.
Great. Well, thank you very much. Thanks to Jen and Cameron for both setting these up and the invite. As well as it sounds like it's going to be an exciting summer with the two talks that have already happened, and so as well as the nice slate of speakers to come. And so I appreciate being offered some time to talk today. So today I'm going to talk about redox transformations, mainly just focusing on a manganese redox transformation, a chromium one that the lab has been working on. But I normally spend a lot of time giving a talk and talking about the how, but today I want to spend maybe a little bit more equal time trying to ponder that why. So why do microbes play a role and express so many unique proteins to participate in some of these redox reactions? And so that's hopefully what I will get across today. So just a rough overview of how I kind of view sort of my work, and it's kind of like microbial geochemistry. And so technically I have a geology background, and so my work kind of looks at microbes as geochemical agents, whether they're looking at mineral dissolution, particularly today, and a lot of my work is looking at mineralization processes, particularly manganese oxides, which we'll hear about today. As well as that microbes also play a fundamental role in elemental cycling in the environment, and today I'll talk a little bit about chromium reduction. And so the cycles that really get me excited and interesting are these coupled biotic-abiotic reactions. And these are where bacteria and kind of the environment and the geochemistry and the minerals are all kind of playing somewhat equal roles together and driving these processes. And so why would you care about minerals and microbes? Well, with their powers combined, they govern a lot of environmental cycles. And so sure, they can deal with these cycles as a microbe and as a mineral, but together they can play a really unique role together, whether or not we're just talking about nutrient cycling, contaminant fate, maybe playing a more indirect role like sorbing on either with like mineral structures or even EPS from bacteria, as well as that minerals can also, and bacteria play, again, these redox cycles, whether it's biotically, abiotically, and also together with one another. So just to hit one more time, the geologist in me wants to just emphasize a little bit the importance of minerals, is that the biogeochemical impact is a function of mineral structure. So not all minerals are the same, and not even all manganese oxides or iron oxides are the same. And so that mineral structure is important to this biogeochemical impact. And so manganese oxides in particular, the mineral that I've spent a lot of time working on, have a very unique structure, and this is an example of that structure, and it's a layered structure that gives it a really good affinity to sorb nutrients like phosphate, sorb contaminants like lead, as well as that the mineral manganese oxide itself, certain forms of this mineral can also play an abiotic role in carbon degradation, as well as it can abiotically just oxidize chromium-3 to chromium-6, which is the opposite of what we'll be talking about today. But this mineral can just play that role by itself. And so, as just a short background, we'll talk about how is this process going on? How is making minerals wrapped with this biological process? And so, an oversimplified view of this is simply just manganese 2, which is a soluble form, and the bacteria then oxidizes manganese 2 to manganese 3, which is also soluble, but it's unstable. And so this manganese 3 will then either be oxidized or disproportionate into a manganese 4 oxide, which is the insoluble form of this mineral. And so I won't give a ton of background, but the one thing that I want to just quick point out is there's a lot of cool and exciting stuff coming out about manganese just very recently. And so if you want more background, there's a great review up top here that I posted, as well as that there's been some exciting stuff in the past year coming out on other systems. And so I'm just going to talk about a organism and how it's oxidizing manganese. But there are other pathways out there, and a more classic example is these multi-copper oxidases. So there's a great paper that just came out. Another great paper talking about the importance of manganese 3 in the environment. May be unstable, but it's very important in terms of the manganese cycling in the environment, as well as that it's not just bacteria. So there's even some cool work coming out. This is from Colleen's lab, looking at fungi and the role they play. And today my work is really mainly focused on a lot of laboratory-based experiments, but I'll also point out that I heard a great talk at AGU this fall looking at metagenomics and manganese cycling. And so that's kind of, not only is there exciting stuff happening and being published right now, but there's exciting stuff on the way. So I really look forward to seeing some of this stuff keep progressing. So particularly, the work that we've been doing is on a unique organism called Rosiobacter, AZWK3b. And it has a very unique sort of story in how it oxidizes because it's a coupled biotic-abiotic manganese oxidation process. And it starts off with the bacteria producing superoxide, oxidizing manganese 2 into these solid-phased biogenic minerals. The really cool thing about this story is it's not just that it produces a biotic mineral, it's that this mineral is actually incredibly reactive and then can further autocatalyze and make a secondary mineral, which I think is cool, again, showing that really tight connection between this oxidative process and mineral formation. And if you look at these minerals, it's even cool that you can even see them in the laboratory. And so that initial biogenic manganese oxide has a different structure and a different reactivity. And so this is what it looks like in a flask. And so as you look under in a TEM, you can see that this initial biogenic manganese oxide is incredibly small. It's not very crystalline, but this is able to autocatalyze and ripen into a secondary phase. And so you have these pathways going on that the biology is making a phase, and then you have geology and the minerals also making a secondary phase, and this secondary phase is less reactive. And so that's why it's important, right? The biotic phase is very reactive, and the secondary phase is less reactive. But what I've spent a lot of time recently in my lab, as well as in collaboration with Colleen Hansel's lab, is looking at this recently. So how is the bacteria doing this process? And so we have some work out there showing that it's superoxide. So the question is, what is the bacteria doing to produce superoxide, and how is this allowing for manganese oxidation to occur? And so some recent work that we're pretty excited about that's been headed up by one of Colleen's postdocs, Pete, as well as I've had a nice undergrad in my lab who's helping out with some of this as well. And we've been able to implicate heme peroxidase in not only manganese oxidation, but also superoxide production. And this was surprising. We kind of thought that we'd find multiple proteins involved in both of these pathways, not necessarily one for oxidation and also superoxide. We thought they would be different. And so the data that I'm showing you right now is just a little piece of it. But you can see that this is a soluble protein fraction. This is a regular Kamasi gel, the boiled and non-boiled sample. And so it's our soluble, it's our protein fraction that's able to oxidize manganese. So evidence of that is the second stain right here, the second gel in the middle. You can see that this is our boiled and non-boiled sample, and we can get manganese oxidation in the gel to occur. Not only can we do an oxidation stain, but we can also look for superoxide production in the gel. And so this is some work that Pete was doing, and able to identify this band. And so in concert, we cut out all these bands, sent them to a mass spec facility, and the band that always dominates is in every band we cut out has these heme peroxidases. So pretty surprising, and hopefully this sort of work will come out soon. And I won't give you a lot more data, but we have a lot of data on this, really showing in multiple different fractions and multiple different assays that this is all pointing back to heme peroxidases. So how is this playing a role in this oxidation pathway? So one possible mechanism for the heme peroxidase to generate superoxide is this. And so it's somewhat of a classic peroxidase reaction by scavenging this hydrogen peroxide and then reducing an organic. What we think may be happening is that that organic may be reducing and getting made into a radical organic. That radical organic can then react with oxygen to form superoxide. And then what happens, and this is more abiotically, but biogenically driven, is that manganese 2 gets oxidized to manganese 3, as well as it generates hydrogen peroxide. And so we've also found out by doing some purely chemical assays is that if you just have superoxide and manganese together, you don't get these minerals. So you have to deal with hydrogen peroxide because that inhibits this reaction of going from manganese 3 all the way to manganese 4. So we think this heme peroxidase could also be playing a role in scavenging or balancing the effects of this superoxide and hydrogen peroxide that allows for the production of these manganese oxides. And so this is still work that's highly underway, and we're still really working on the how and how this really balances out. And we were lucky enough to get some funding from NSF to continue this work to try to define this indirect pathway for bacterial-driven manganese oxide formation. And so right now I have 3 students in my lab, Isaiah Reynolds, Sarah Phillips, and Jim Dunn, kind of looking at protein expression of these heme peroxidases in multiple conditions, and also trying to purify these heme peroxidases to try to see if we can further prove their function in this role. So it brings me to my next question, the one that I don't think I get to talk about all too much is the why. And I think this is one of the coolest things about manganese oxidation, especially for bacterial manganese oxidation. And so currently, there's no real known physiological reason that works for all organisms on why they oxidize manganese. For the most part, there's not really great evidence that shows energy is conserved. And so if energy is not conserved, why would an organism do this? Why would they make a big heme? Why would they make other proteins that are able to do this? And so some proposed reasons that are out in the literature is maybe that the idea for an organism to make these minerals is to store electron acceptors. Maybe the production of manganese oxides, because they're great scavengers of contaminants, maybe that sequesters these contaminants, allowing for the bacteria to survive. Maybe they break down complex organics, making more labile carbon for the microbes to utilize. Another possibility is maybe degradation of reactive oxygen species. However, in terms of Rosiobacter, if we specifically ask the question of why does Rosiobacter do it? Well, at the moment, storage of electron acceptors, we're just going to cross off the list because it doesn't reduce. It's not an anaerobic organism. And surprisingly enough, even though reactive oxygen species is involved, the presence of manganese doesn't help this organism survive and live at the moment that we have data on. And so we don't really think it's a degradation of reactive action, this ROS, or oxidative stress response. And so we're really stuck with maybe a couple of the proposed reasons or just a question mark of what. So to try to address this, we tried to look at Rosiobacter's protein response to manganese 2. And so we grew up Rosiobacter without and with manganese chloride. And then we let it grow up, we extracted the proteins, and we tried to analyze its active portion of the protein, so its soluble fraction, in 2 different growth conditions using gel-CMS. And so it's just essentially running this one-dimensional gel, cutting up into fragments, and those fragments are then ran on an LC-MS, and then we get the proteome back. And so to try to make a short story out of this is, with the data from this experiment, we saw that Rosiobacter actually had a surprisingly small overall response to manganese 2. And so this is the entire data set that you're looking at right now that we've been able to functionally characterize and cluster into different groups. And at the moment, the differences between, on a very big, broad global level, is very small. And there's not necessarily a connection that, well, metabolism, manganese is always up. Manganese always causes an increase in expression over all these functions. And so it kind of left us a little bit puzzling, wanting to know more about this. And so, but the more could also be related to its isolation condition. So this is an organism that it was isolated with manganese. And so maybe the surprise of having manganese or not manganese is not something that we should have expected a large proteome response just because of how it's normally grown and how it's isolated. So what did this assay, what did this experiment tell us? Well, the one thing that it did tell us is that Rosiobacter does have a requirement for manganese too. The one thing that was very clear within the proteomics data is that when we looked, not on a broad level, but we sifted through the data, the 2 or 3 proteins that were significantly expressed in the presence and absence of manganese were manganese transport proteins. And so when there's a lot of manganese, the bacteria didn't express the transport proteins. But when we didn't add manganese, they expressed the transport, and they're trying to scavenge to get manganese from the environment. And so that's the one thing we did learn. We were surprised that there was no differential expression with any proteins really related to oxidative stress, superoxide production, maybe NAD oxidoreductases, as well as our hemes or our manganese connection proteins. We didn't really see that response. So if Rosiobacter isn't really responding and making oxidative or oxidative stress response or manganese oxidation connections based on the presence or absence of manganese, it kind of makes me ponder, what if oxidation by Rosiobacter is indirect? What if the oxidation may be happening as a side reaction due to metabolism from this organism? It's going along, it's oxidizing, but it's mainly dealing with carbon, nitrogen, metabolizing, and it just so happens to be producing some side products that allow for oxidation to continue. Again, this is I will say that lots of folks don't like the idea of the bacteria kind of not doing this for a reason, but at the moment it's something fun, and like I said, I'd like to ponder about that question. And also there was a study that just came out in AEM, and maybe even within a couple weeks ago, looking at manganese oxidation and looking at low pH environments. And they also found 2 or 3 different isolates that are able to oxidize, but if you change the pH, the growth conditions, then all of a sudden oxidation is shut down. And so this maybe even, if I were to speculate on some of their results as well, maybe makes me ponder that as well. So are these organisms maybe doing it indirectly oxidizing? Maybe the change in pH is just causing a physiological change that then cuts off a metabolic pathway that the organism is no longer using, thus maybe cutting off oxidation. So the pondering of the why is something that I will continue to ponder, and I think it's a great question for the manganese field, and I think it's a very important one to think about. And so now we're going to make a somewhat of a crude transition, because now it's no longer manganese, it's no longer oxidation, but now it's going to be about looking at chromium-mediated, microbial-mediated chromium reduction. And so again, I'd like to oversimplify our pathway here, and so we have chromium-6, which is a carcinogen, it's a toxin, it's very soluble. However, microbes, minerals, they can play a role not only in the abiotic oxidation, but also in the reduction into chromium-3, which is a less toxic form, as well as it's a less soluble form. And so this is a favorable reaction in the environment. And I'll point out that this was work done by my now former master's student, Mike Henson. And so any complaints can just be sent directly to his Twitter account right there. So I will definitely point that out. And so today what we're going to talk about and how we started this is so looking into this pathway. And so at first I think when I tell people that we're looking at chromium reduction, they say like, oh, well, yeah, that's fine, but that kind of sounds boring. We have it all figured out, and so, okay, let's just move on. However, I thought the same thing too. As well as, so when we started this project, I remember when Mike was in the office and we were trying to discuss where to go with this project, we're just like, well, this should be a little bit more straightforward. Chromium reduction should be more straightforward. And the one thing that I want to point out is we're not talking about anaerobic reduction and using chromium as a terminal electron acceptor. We're just talking about aerobic reduction. So the organism is growing in oxygen, but just able to reduce and resist hexavalent chromium. And so this should be more straightforward. It's a clear toxin. Manganese, not necessarily toxic, right? When you think about hexavalent chromium, it's clearly a toxin. So we'd expect an obvious response from a microorganism. Not to mention that the resistance gene, an efflux pump, if you look into the databases, there's almost 10,000 protein annotations based on this efflux pump. And if you look for reductases in NCBI, you'll see that there's almost 6,000 proteins. So we said, okay, so now we have some genes, let's track some expression, then we can go to the environment, because that's where I've always wanted to go, even with the manganese. We want to try to test it in the lab and then go to the environment. But a lot of times we're stuck wondering, what are we testing? What genes are we looking for? And so essentially we started this project thinking, well, this should be easier. We'll find these genes and then we'll start playing around. So the project started out, we had a bunch of isolates in the laboratory that were actually isolated by Peter Kortev, who's a colleague of mine at CMU. And so we decided to look at 4 environmental isolates that were chosen based on differences in growth and chromium reduction. So we wanted a nice comparative set. And so this is some work that Mike is working on writing up right now. And so the next data that I'll be showing you will be in that manuscript soon. So you can see that we have 4 isolates, 2 that are very fast reducers, and then 2 that are slower. Okay, and so we thought this would be a very good comparative set. So we could say, like, well, we have 2 fast ones and a slow one. So now we can look for those genes and we'll see what happens, and we should have this clear idea of reduction. As well as the other cool thing about these organisms, you can see that they also have different signatures of their growth in terms of their growth curves. And so our fast reducers are fast growers. Our slowest reducer is not actually our worst grower. And so this is our slowest reducer. So we have some variations, but we were looking at these and we're like, all right, this is a good subset, and so now we dive in. Well, something that already started to throw a little bit of a wrench into our system is that surprisingly, all 4 of these isolates were Microbacterium species. So at the 16S level, we had, I think, around 1,400 base pairs, and they were 100% identical. So we were surprised about that. We were kind of thinking we'd get at least some variation in terms of maybe genre or OTU classification or taxonomically where they broke down. And you can see that those are our fast reducers and these are slower reducers. And so this brings me back to Mike's talk from a week or 2 ago. And I really thought this figure that he showed and presented was really interesting because it looked at the impact of changing diversity on geochemistry. And I think there'll be maybe a handful more talks like this. And I think this figure was great because it really begs that question of what is the change in this OTU richness and how is that impacting the geochemistry? Which is a great question. And how are the microbes responding? Is it taxonomic difference? Is it genomic difference? So it's a great figure to ponder on. And also, if you haven't watched that seminar, you could go to the website down here and you can go catch up because it was a great talk. However, at least for the chromium reduction work, we saw that the OTU technically would be the same in the environment, but they have different geochemical potentials. And so this is why I had an affinity for this figure that Mike presented, because it's been something I've been thinking about only recently. So what we decided to do is then try to evaluate the genomes. And so the genomes were sequenced and then assembled. This is all done by my student, Mike Henson, so he deserves a lot of credit for that. And so the really cool thing is that not only geochemical ability to reduce chromium was different, but we also saw that these genomes showed strain variation between the isolates. And so here are slower reducers, and here are more faster reducers. And you can see big pulls, and this probably is no surprise, right? Because these are just PFAM categories, presence, absence, a PCA that you're seeing right here. And you can see that carbohydrates metabolism, inorganic ion transport metabolism, transcription, and then the doomed unknown function, right? It's really tough to say a lot about those unknown functions. However, we were surprised by this variation. Since metabolism in the genome was different, we decided to also do some metabolic fingerprinting to show further variation within these organisms. And so, to no surprise, especially if you consider the old-fashioned growth curves, remember that these two, you may forget, but these two grew very similarly in terms of their growth curve. And it just so happens their metabolic fingerprint, their ability to oxidize a variety of carbohydrates, amines, polymers, amino acids. These are very similar, and then these two had more unique growth patterns, and so their metabolic fingerprint also shows greater variation. So, what about chromium-related genes? Okay. Within our annotations and assembly, we did not see any presence of an efflux pump. Okay, so that was surprising. Okay, remember, I want to emphasize that this was supposed to be the simple story. And so we found that there was no real efflux pump present that was specific to chromium relative to what the literature has shown. The other thing that was surprising is that our automated annotations, whether or not it was IMG, and we also did an annotation with RAST, but we never really saw the presence of a chromate reductase, that CHRR, YEF, via those automated annotations. So we decided to dig a little deeper, and Mike ended up doing a manual database that he made from Uniprot data. And he did end up finding some candidates for the CHRR and YEF, which are both broad category oxidoreductases. Okay, you can see that the good news is our CHRR did have some sequence similarity based on an organism that has shown the ability to reduce chromium. And so we do have some potential, some candidates that we really have to evaluate to see about their role in chromium reduction. So, why is reduction so different? Is reduction different because of the presence or absence of genes? Well, I can't really say yes or no, but there's no obvious reason right now. It's not as though our fast reducers have a certain group of genes that we've been able to identify that really link it to survivability, oxidative stress, or anything like that. So at the moment, there's no obvious yes. So at the moment, it's kind of a no. Is reduction different between these strains because of the expression of these reductases that are maybe candidates? This is something we are evaluating right now with qPCR. And so this is something that's still in the laboratory getting worked on. However, at least at the moment with the literature looking at chromium reduction, there's no real great connection through these isolates and organisms that have been looked at in terms of the presence of chromium and the expression of these reductases. And so, while we're still going to look into that, we're not really sure if this is going to come about. And so, as you can see, maybe the pattern that's coming along, based on my indirect comments earlier. So now we're kind of wondering for these organisms, is the reduction differences connected to metabolism? They're able to metabolize different types of carbon, and so maybe is that metabolic potential related to its ability to reduce and resist chromium? Why we think that is because chromium toxicity, hexavalent chromium toxicity, is relating to exhausting reducing power of the cell. And so if organisms have a way of metabolizing, fighting against that exhaustive reducing power, maybe that allows them to survive and reduce the stress related to chromium, hexavalent chromium. And so this is definitely scratching the surface and really trying to ponder these questions of the why. And so we're not really sure, even though this was supposed to be our easy answer of why this is happening, why they're different. And so today, as I wrap up, I'll just say that so what we try to do is we try to look at these mechanisms, and a lot of my work is done in the laboratory, looking at test tubes, trying to define these mechanisms. We are aware that the environment exists outside of the test tubes. We're trying to get there, and we do have some projects that are just undergoing right now, looking at some wetlands within the Great Lakes and some Antarctic sediments that a colleague at CMU collected for me, Andy Mahon. And so we're trying to connect these two areas from the test tube to the environment, but trying to identify these genes and these chemical pathways have been something that's been a little more tricky than we've assumed. And so with that, I'll just wrap up, but this is the lab. I've got a great group of students at the moment. One is just leaving, and I have a couple more coming in, so if there are any interested students, we're always looking for more people excited about looking at these coupled reactions. And so with that, I'll just go ahead and acknowledge NSF for some manganese funding, support for some other projects, as well as collaborators like Colleen Hansel, Andy Mahon, Jorge Santo-Domingo, and others. So with that, I wish you all a happy 4th of July, and let me know if you have any questions.
Thanks, Deric. That was awesome. You're really making the effect going, right? So what we can do is let's take questions from people in Google Plus right now, and if everyone could keep their microphones muted unless you're speaking up. Does anyone have a question right now that's online with us? Henson's not allowed to ask.
I've got a question.
All right, Mike, go.
So, the 4 microbacterium species, at what point does chromium become completely toxic to those species and sort of actually inhibit growth?
That's a great question. So that's some data that I didn't show. And so the 2 fast reducers can survive in up to 100 millimolar chromium-6. The other two lower reducers only survive around 10 to 5 millimolar chromium-6, which millimolar concentrations of chromium is still a really large amount. And so the organisms that are able to reduce faster survive up to 100, but over 100 they kind of die off. And the slower ones die off around 5 to 10 millimolar, excuse me.
Like you say, that's still pretty impressive, though.
Yeah, yeah. Even our slow reducers are pretty resistant to chromium.
Yeah.
Yeah.
I have a question. Mike, are you done?
Yeah, I'm done, Cam.
Do you know that the chromium is getting into the cells, or how much is getting into the cells? I mean, is it something that can passively diffuse in or go in through porins, or does it need active transport, or how? Like, I guess I'm just wondering with this toxicity effect, how much might be entering the actual cells themselves and interfering with things once it's in there?
Yeah, no, that is a great question. I think the chromium can transport in. And right now we don't really have any data on that at the moment, and so we haven't really got to the fractioning out of cells. But there have been a few studies that have looked at that. But that's how they get to that reducing pool by that chromium-6 getting in there and then being reduced to chromium-5. And so that ability to then transport in, so that's where some of the toxicity does generate, because it's getting to that NADH pool, that reducing pool.
All right, Minnesota, you guys want to go live and chat with us?
I'm muting. There. All right. This is Minnesota. I have a manganese question, and I apologize if I missed this earlier. Is the protein associated with the manganese oxidation induced by the presence of manganese in your proteome experiments, or is it constitutive?
No. So from the data we have thus far, we have seen no evidence of any expression based on manganese. It's constitutive. We are looking into that within Western blot experiments right now. So the issue is that Rosiobacter has 3 different heme peroxidases that are almost identical. And so when we see it in the mass spec, it all just gets mapped back to the dominant isoform. And so it's really tough to say that, well, is it the big, middle, or small one, because it's all very similar in terms of its amino acid sequence. And so at the moment, we don't have evidence to say that it's differentially expressed based on manganese. However, that's something that we're working on right now, and an undergrad in the lab has been working on Westerns for almost 6 months, trying to transfer 230 kilodalton proteins over, and then trying to get the binding to affinity binding to work out. So.
And one follow-up: when they produce the solid manganese, do they associate with it exclusively? Do they basically sync with it?
No, they don't. And so this is an organism that's able to oxidize when it's growing. So in early stages, in exponential phase, so this organism is actively oxidizing and growing at the same time, and it's producing extracellular manganese oxides outside the cell.
All right. There goes one great idea. Another one down.
All right.
All right, any other questions from the Minnesota beer drinkers? Jeff on his cell phone.
Yeah. I think he's Jeff missed it. So roll call. Any questions?
All right, anyone else on Google Hangouts want to unmute yourself and ask a question? Just a reminder, if you're not in the screen right now, we need you to do it through Twitter because we can't hear you. Okay, well, I had a question for Deric, actually. When you did the comparison of the IMG and RAS pipeline and saw no potential homologs, but then got it through UniProt, and those are all both like homology-based searching, so what was the major difference between the two categories or databases in terms of being able to pull it out of UniProt and not the other ones?
Yeah, I think the UniProt was just a little broader, and the tightness of the call was much broader with that, because we were able to curate it a little bit. And so the IMG would have been a little bit more specific based on the CHRs that have been looked at in the laboratory with like Pseudomonas. There's been a lot of great Pseudomonas E. coli that have looked at that sort of reductase. And so the UniProt ended up being more of a broad general oxidoreductase, and we ended up finding one that was connected to a chromium reducer, so broader. Yeah.
It's a little frightening to think you guys are pointing out, right, how much we can miss with our database searching in terms of a lot of
So
functionality.
it's been really surprising to me as well. I've joked that we should have error bars on our genomes and our assemblies, which yeah.
I wanted to bring up a comment here for these kinds of things. There's a guy here at LSU named Microbial who does structural prediction, homology searching, and it's probably outside the scope of your current work. But if you want to try and screw around with another way of finding potential homologs, they have ways of comparing the structures, predicting the structures of the binding pockets, the potential ligands, and finding things that might be homologous at that level. And I can either get you in touch with them or show you their software or whatever. It's another way to it's another road to run down, and it's a lot. There's a lot more manual effort involved because of the fact that it's so much. There's so much evaluation of the results with the potential predicted sort of targets, but it might be something that could get you a few more things to try and chase after if you need some.
Yeah, no, thank you. That's a great idea. Yeah, we're always looking to add more candidates to the list. The tough thing is then evaluating all the candidates in terms of getting that function connection. So.
There's a question just came in on Twitter.
Yeah, I'll let Cameron do it then.
Oh, sorry. It's from Paul Carini. He asked if you looked for manganese riboswitches in the genome.
Manganese riboswitches. Wow. No, we certainly have not done that. That's something that we certainly could add to our list, but that's not one thing we've even pondered, to be honest. So, okay.
Paul, if you're listening, do you know how many different types of metal-activated riboswitches are out there? Do you know kind of what range of things we're talking about there? Other examples are there iron riboswitches, or I sort of think of them as activating by small molecules, but not necessarily by sort of single atoms or minerals. You can throw us one on Twitter while we talk about something.
Yeah, okay, now we'll see how long it takes for him to type.
Yeah, the one thing that even within some of the other manganese model organisms that are out there, that connection with manganese, the presence of manganese and the expression of genes related to oxidation, there's not really any evidence that's shown really great connections between those. So that's one of the issues with this manganese conundrum as well. So it's not necessarily just responding to the presence of manganese and turning on a system that makes these oxides.
Well, Paul just wrote back in as a fluoride riboswitches, so potential that metal could, I guess, be activated, but very interesting to think if there's specific ones or general ones for metal ions. Anyway, well, with that, anyone else have any questions? My Wi-Fi is updating. But if not, I'll remind you that we'll have a talk in 2 weeks on Friday. We're going to switch back to Friday since we're done with holidays. And check the website for updates, and we'll keep adding to the schedule. And thanks to everyone for logging in. And hope if anyone's on the Atlantic coast along with me that we ride out the storm safely. So I think everyone have a great day and enjoy your holiday weekend. And thanks, Deric, for doing the seminar.
Yeah, no worries. Thank you very much for the offer. Thanks for everybody taking some time on the day before the 4th of July. So.
Great. Thanks, Deric.
Thanks.
Cheers.
Thanks, everyone.